Hi, good morning, students. How are you? Habiswas family members, I believe you all are fine and safe. Okay, this week we are going to discuss the topic DNA metabolism in plants. Understand, students? In the previous day, we studied about the genetic mapping, isn't it, students? Okay, what is the DNA metabolism in plants? What is DNA replication? In the double helix, the two parental strands of DNA separate, and each parental strand synthesizes a new complementary strand. Okay, listen well. In the double helix, two parental strands of DNA separate and synthesize the parental strands to new complementary strand. Okay, the DNA replication is semi-conservative. Okay, therefore, each new DNA molecule conserves one original strand. Okay, that is known as DNA replication. Okay, next, what is DNA repair? What is DNA repair? The DNA repair system plays a major role in maintaining the genomic or genetic integrity of an organism. It will maintain the genomic or genetic integrity of an organism. Understand, students? The DNA repair system protects the integrity of genomes from genotoxic stresses. Okay, இந்த மாதிரி genotoxic stressesல இருந்து ஜினோமிட்டியா ப்ரொடெக்ட் பண்ணக்கூடிய ஃபங்க்ஷனை பெர்ஃபார்ம் பண்ணிக்குது. ஓகே, நெக்ஸ்ட் we want to discuss about the what is OR. What is OR? Okay, yes, that means what is origin of replication. Okay, replication stores at the specific site on a DNA sequence. The replication ஆனது specific site of DNA sequencesல start பண்ணுது. That is known as origin of replication. Understand, students? What is origin of replication? Replication starts at the specific site on a DNA sequence known as what? Origin of replication. Understand? Next, we want to study about the what is ARS. What is the full form of ARS? Autonomous replicating sequences. Okay, the replication process, the origin of replication takes place in yeast is called ARS. That means autonomously replicating sequences. Understand, students? Then next we want to discuss about the protein synthesis in plants. Okay, so they are first definition, transcription. Okay, that takes place in two ways, transcription and translation. Understand? What is transcription? Transcription is the process in which one strand of DNA acts as a template. One strand of DNA template act panni to generate mRNA. What do you mean by mRNA? To generate messenger RNA. Okay, to generate messenger RNA with the bases complementary to the template strand. It is catalyzed by the enzyme called RNA polymerase. Okay, so in the enzymes nala idu catalyzed agudhu RNA polymerase nala. Okay, students, next we want to study about the template strands or non-coding strand and antisense strand. இந்த மூணுல எது கேட்டாலும் கரெக்ட் ஆன்சர், ஒரே ஒரு ஆன்சர்ஸ் கரெக்ட். ஓகேயா, மூணுத்துக்கு ஒரே டெஃபினிஷன்ஸ் தான் இருக்குது. சரியா பிள்ளைங்களா? ஓகே, த ஸ்ட்ராண்ட் ஆஃப் டிஎன்ஏ விச் இஸ் ஓரியன்டட் இன் 3' to 5'. அப்போ டிஎன்ஏவில் ரெண்டு ஸ்ட்ராண்ட்ஸ் இருக்கும், 3' to 5'. ஓகே, ஸோ 5' to 3'. 3' இருந்து அந்த ஸ்ட்ராண்ட்ஸ் ஆஃப் டிஎன்ஏ விச் இஸ் ஓரியன்டட் இன் 3' to 5' direction that serves as the template for the synthesis of messenger RNA. Understand? Very easy, no? That is called a template strand or non-coding strand or antisense strand. Next, what is coding strand or non-template strand? Okay, the other strand of DNA which is transcribed. Other strands of DNA which is transcribed is called coding strands. Okay, next one, what is promoter? The specific sequence of DNA nucleotide called the promoter. This is essential for transcription. The promoter is எதற்கு மிக தேவையான ஒன்றா நெசசரியாக இருக்குது transcription processக்கு promoter very very necessary. Understand students? Next definition, what is TATA? What is TOTA or Hogness box? The transcription stored site contains about 25 BP, means base pairs upstream. The sequence is TATA is known as TOTA or Hogness box. Okay, that definition also you want to study well. Next, very very easy definition. What is heterogeneous nuclear RNA? The RNA polymerase transcribes the precursors of messenger RNA, which are also called as heterogeneous nuclear RNA. That means XII RNA. H refers to heterogeneous, N refers to nuclear. Understand students? Okay, next, what is capping? Modification at the 5' end of the primary RNA transcribed with the methylguanosine triphosphate. Methylguanosine triphosphate is called a capping. Okay students. Next, what is the purpose of capping? Protect RNA from degradation. Capping plays an important role in removal and first intron in the pre-messenger RNA. It regulates it regulates the messenger RNA export from the nucleus into the cytoplasm. It helps in binding a messenger RNA to the ribosomes. Next one, what is tailing? The 3' end of heterogeneous nuclear RNA is cleaved. அது cleaved ஆகுது. எந்த enzymes-னால cleaved ஆகுது? Cleaved by an endonuclease, and the string of adenosine nucleotide is ordered to the 3' end of heterogeneous nuclear RNA is known as polyadenylation or tailing. Okay. Next, what is the purpose of tailing? Transition of RNA transcript is facilitated. Second one, helps in the synthesis of polypeptides. Polypeptides ஓட சேர்க்கைக்கு help பண்ணிக்குது. Okay. Then it enhances the messenger RNA stability in the cytoplasm. Okay. இந்த தூதுவர் messenger RNA-ல ஒரு ஆர்கானிசத்தில் இருக்கக்கூடிய சைட்டோபிளாசத்துக்கு ஸ்டெபிலிட்டி நிலைப்பு தன்மையை என்ஹான்சஸ் பண்ணிக்குது. Okay. Next, what is exons? What is introns? Coding sequences, non-coding sequences. Okay. What is spliceomes? RNA splicing is a process which involves the cutting or removing of introns and knitting of exons. This process takes place in spherical particles. In the process, cutting, knitting பண்ணக்கூடிய exons, introns, அந்த process ஒரு spherical particles-ல நடக்குது, which is a multi-protein complex that is called as spliceomes. Okay students, this much you want to study, write and send to today itself. Okay, without fail. Okay, God bless you all. Thank you students.